Welcome to this lecture on cell and molecular biology. Today, we are diving into a truly fascinating topic, the charging of tRNA. If you have ever wondered how your body flawlessly translates genetic code into the physical proteins that keep you alive, it all starts right here. This process is the crucial bridge between the information in your DNA and the actual construction of proteins. By the end of this video, you will understand not just what this process is, but exactly how it works, why it requires so much energy, and how it maintains such incredible accuracy. Let us get started. To understand what is happening at the molecular level, let us imagine you are running a massive construction site. You are building a complex skyscraper, which represents a protein in the cell. To build this skyscraper, you need specific building blocks, which are the amino acids, delivered in an exact, predetermined order. The delivery trucks that bring these blocks to the site are specialized molecules called tRNA. However, a delivery truck is completely useless if its cargo bed is empty. Before a truck can drive to the construction site, it must first go to a loading dock to pick up its specific cargo. In the cell, this crucial loading process is called the charging of tRNA. Now that we have our analogy, let us look at the formal biological definition. In molecular biology, the charging of tRNA is formally known as amino acylation. It is a highly specific two-step biochemical reaction where an amino acid is covalently bonded to the 3' end of its corresponding transfer RNA molecule. This reaction does not just happen on its own. It is catalyzed by a specialized family of enzymes called amino acyl tRNA synthetases. There is typically one specific synthetase enzyme for each of the 20 standard amino acids found in living organisms. The enzyme ensures that the correct amino acid is matched with the correct tRNA, which is identified by its specific anticodon sequence. Understanding the definition leads us to ask why this specific step is so vital. The charge-in of tRNA is arguably the most critical quality control step in all of gene expression. Why? Because the ribosome, which actually builds the protein, is completely blind to the amino acid itself. It only reads the tRNA anticodon to decide which block comes next. If a tRNA is accidentally charged with the wrong amino acid, the ribosome will unknowingly insert the wrong building block into the growing protein chain. This would lead to misfolded, non-functional, or even toxic proteins, causing severe diseases. Furthermore, this charging process stores vital energy in the chemical bond between the amino acid and the tRNA. Knowing that this process requires energy, let us examine the chemical equation that makes it happen. The charging of tRNA is an endergonic process, meaning it requires a significant input of energy, provided by ATP. The equation shows amino acid plus tRNA plus ATP reacting to form amino acyl tRNA, AMP, and inorganic pyrophosphate. Let us break down these symbols. AA represents the specific amino acid. ATP is adenosine triphosphate, the universal energy currency of the cell. On the product side, AA-tRNA is our final charged delivery truck, ready for the ribosome. AMP is adenosine monophosphate, what remains after ATP loses 2 phosphate groups. Finally, PPI is inorganic pyrophosphate, which is immediately broken down to release energy, driving the entire reaction forward irreversibly. This chemical equation we just saw leads us to an important question about energy consumption. How much energy does a cell actually spend to charge these molecules? We can calculate this using a simple formula. B equals N times 2. In this equation, the variable B represents the total number of high-energy phosphate bonds that are cleaved during the charging process. The variable N represents the total number of amino acids that need to be charged onto tRNAs to build a specific protein. The number 2 represents the two high-energy phosphate bonds that are broken when one single ATP molecule is reduced to AMP. If the length of the protein increases, the energy cost increases proportionally. Before we move on, let us see this formula in action with a concrete example. Imagine a cell needs to synthesize a small polypeptide chain that is exactly 40 amino acids long. We're asked to find how many high-energy phosphate bonds must be hydrolyzed just to charge the tRNAs required for this polypeptide. We start by identifying our given value, which is N equals 40. We then use our formula, B equals N times 2. By substituting 40 for N, we multiply 40 by 2. This gives us our final answer. Exactly 80 high-energy phosphate bonds are consumed. This tells us that the cell invests a massive amount of energy just preparing the building blocks before assembly even begins. Now that we have used this formula, let us understand exactly where it comes from. Why do we multiply by 2? We must look at the stoichiometry of the tRNA charging reaction. For every single amino acid that is attached to a tRNA, exactly one molecule of ATP is consumed. However, this ATP is not just converted to ADP, it is converted all the way down to AMP and inorganic pyrophosphate. This specific conversion effectively breaks two high-energy phosphoanhydride bonds within that single ATP molecule. Therefore, the energetic cost per individual charging event is equivalent to 2 bonds. To find the total cost, we simply multiply the total number of amino acids by 2. We have established the energy cost, which brings us to another critical factor, accuracy. Because charging is the ultimate quality control step, we need to know the probability of building a completely perfect protein. We use the formula P equals the quantity 1 minus E raised to the power of L. Here, the variable P represents the overall probability that the entire synthesized protein has absolutely no errors. The variable E stands for the error rate of the enzyme per individual charging reaction. The exponent L represents the total length of the protein, measured in the number of amino acids. Notice that as the length L increases, the probability of a perfect protein decreases exponentially. Let us apply this accuracy formula to a realistic biological scenario. Suppose an amino-acyl tRNA synthetase has an error rate of 0.0001, meaning it makes one mistake in 10,000 events. We want to find the probability of synthesizing a completely error-free protein that is 300 amino acids long. We are given E equals 0.0001 and L equals 300. We substitute these values into our formula. 1 minus 0.0001 gives us 0.9999. We then raise this to the power of 300. The final answer is approximately 0.9704. This means there is a 97% chance of producing a perfect protein. So we have successfully used the probability formula. This brings us to understanding its mathematical foundation. We are setting out to derive why the formula takes this specific exponential form. We start by looking at a single charging event. Let the variable E represent the probability of an error, or an incorrect charging event. In basic probability theory, the sum of all possible outcomes for a single event must equal exactly 1. Therefore, To find the probability of a perfectly correct charging event, we simply subtract the error rate from 1. This gives us the expression 1 minus e. This represents our baseline success rate for attaching just one single amino acid correctly. Continuing from our single event, we now extend this logic to the entire protein chain. A complete protein requires L consecutive successful charging events. A crucial biological fact is that each charging event is statistically independent. The enzyme does not remember its previous mistakes. In probability theory, to find the likelihood of multiple independent events all happening successfully, we must multiply their individual probabilities together. So we multiply 1 minus epsilon by itself, repeating this multiplication L times. Mathematically, repeated multiplication is expressed as an exponent. This directly yields our final result. P equals the quantity 1 minus epsilon raised to the power of L. This highlights why enzymes must have incredibly low error rates. Having proven the mathematics behind the process, let us visualize the actual physical mechanism. Here, you are looking at a schematic diagram of the aminoacyl tRNA synthetase enzyme, shown as the large blue structure. On the far left, you can see the two initial reactants entering the enzyme: the amino acid, represented by the red circle, and the ATP molecule with its 3 green phosphate groups. Following the arrows to the middle, we see the first step. The amino acid reacts with ATP to form the intermediate aminoacyl AMP, while 2 phosphate groups are released. Finally, on the right, the uncharged tRNA enters. The enzyme transfers the amino acid onto the tRNA. The fully charged aminoacyl tRNA and the remaining AMP molecule then exit the enzyme, ready for protein synthesis. We have covered a lot of ground today, so let us summarize the most important ideas. First, the charging of tRNA, or aminoacylation, is the foundational step that bridges the genetic code with actual protein synthesis. Second, remember that specialized enzymes called aminoacyl tRNA synthetases are responsible for meticulously attaching the correct amino acids to their corresponding tRNAs. Third, this is an energy-intensive process. It consumes the equivalent of 2 high-energy phosphate bonds from ATP for every single amino acid charged. Finally, and perhaps most importantly, this process acts as the ultimate quality control checkpoint in the cell, ensuring the ribosome builds functional, life-sustaining proteins. You now have a complete understanding of tRNA charging.